Cancer, a formidable foe, is constantly evolving, and in the realm of gynecological malignancies, a fascinating process called lactylation is emerging as a key player. This isn't just about the disease itself, it's about how our bodies respond at a cellular level, and how we can potentially fight back. Let's dive in. For years, lactate was dismissed as a mere metabolic waste product. But now, research is unveiling its pivotal role in regulating cellular functions and, crucially, in the progression of diseases like ovarian, cervical, and endometrial cancers. In these gynecological cancers, the accumulation of lactate triggers lactylation, a unique post-translational modification. This modification acts as a crucial link between metabolic reprogramming and epigenetic regulation, fueling tumor growth, spread, metastasis, and resistance to treatment. The exciting news? New therapies that target lactate production, transport, and lactylation itself show real promise in combating these cancers, especially when combined with immunotherapy. Lactate's role, beyond waste. So, what exactly is lactate, and why is it so significant? Lactate is primarily generated through glycolysis, particularly when oxygen is scarce or when cancer cells exhibit the Warburg effect, a metabolic shift where cells favor glycolysis even when oxygen is available. But lactate does more than just provide energy. It influences redox balance, fatty acid synthesis, and the function of immune cells. In the tumor microenvironment, TME, the accumulation of lactate, often exacerbated by metabolic disorders, contributes to immunosuppression and chemoresistance. The science of lactylation. Now, let's explore lactylation. This is a reversible epigenetic modification where lactate attaches to lysine residues on histones and non-histone proteins. Discovered in 2019, lysine lactylation, KLA, is controlled by a team of enzymes. Writers, like P300/CBP, which facilitate lactylation. Erasers, including DEX and sirtuins, which remove lactyl groups. Readers, such as SMARCA4, which recognize lactylation sites and mediate downstream effects. Alanine tRNA synthetases, ARS1/2, act as intracellular lactate sensors and lactotransferases, promoting lactylation of targets like P53, which is linked to a poor prognosis. Lactylation affects both histone proteins, e.g., H3K18-LO, H4K12-LO, and non-histone proteins, influencing transcription, DNA repair, autophagy, and metabolic enzyme activity. Lactylation in gynecological cancers, a closer look. Lactylation drives malignancy and therapy resistance in gynecological cancers through various mechanisms. Ovarian cancer, OC. H3K18-LO upregulates CCL18, which helps M2 macrophages to polarize, aiding immune evasion. Lactylation of PFKP enhances glycolysis through PTEN modulation. RAD51 lactylation improves homologous recombination repair, leading to platinum resistance. H4K12-LO activates RAD23A and MYC, driving resistance to niraparib. Cervical cancer, CC. H3K18-LO upregulates GPD2, promoting M2 macrophage polarization. DCBLD1 lactylation stabilizes glucose 6-phosphate dehydrogenase, activating the pentose phosphate pathway. HPV16E6 inhibits G6PD lactylation, altering metabolic flux. Endometrial cancer, EC. H3K18-LO promotes USP39 expression, stabilizing PGK1 and activating the PI3K/AKT/HIF1 alpha pathway. PFKM lactylation enhances glycolysis and invasiveness. Targeting lactylation, potential therapies. Researchers are exploring various therapeutic strategies to disrupt lactate metabolism and lactylation to reverse the harmful effects on the body. Metabolic interference. 2-deoxy-D-glucose, 2-DG, inhibits hexokinase, reducing lactate production. Tensionone I downregulates glycolytic enzymes, e.g., LDHA, PFKP, and lowers H3K18 law levels. ENO1 monoclonal antibodies block glycolysis in cervical cancer. Lactate transporter inhibitors. Sarosingapine and AZD3965 inhibit monocarboxylate transporters, MCT1/4, disrupting lactate shuttling. CD147 regulates MCT membrane localization and is a potential drug target. Immunotherapeutic strategies. Lactate-driven lactylation upregulates PD-L1. Combining LDHA or MCT inhibitors with anti-PD-1/PD-L1 antibodies enhances efficacy. Resveratrol inhibits glycolysis and lactate production, reducing TREG-mediated immunosuppression. Other therapeutic targets. LDHA inhibitors, e.g., oxamate, induce apoptosis and autophagy. Metformin, alone or with nelfinavir, reduces lactylation via SIRT3 activation. Cold atmospheric plasma, CAP, downregulates USP49, enhances HDAC3 degradation, and promotes P53-mediated ferroptosis. The road ahead, challenges and opportunities. While the preclinical results are promising, several hurdles remain. One major concern is the broad physiological role of lactate, which raises questions about drug specificity and potential toxicity. Many agents targeting lactylation are still in early development, with few reaching clinical trials. Furthermore, isoform-selective inhibitors and tissue-specific delivery systems are needed to minimize off-target effects. Future research should focus on identifying gynecological cancer-specific lactylation sites using multiomics and machine learning, developing lactylation-based prognostic models and biomarkers, exploring direct targeting of lactylation enzymes, writers, erasers, readers, validating combination therapies in large-scale clinical trials. Conclusion. Lactylation represents a crucial link between metabolic reprogramming and epigenetic regulation in gynecological malignancies. It impacts vital processes like immune evasion, DNA repair, and chemoresistance. Targeting lactate metabolism and lactylation through metabolic interference, transporter inhibition, and immunotherapy offers a promising multimodal approach. But here's where it gets controversial. The complexity of the metabolic pathways and the potential for off-target effects demand careful consideration. Future efforts to improve drug specificity and validate lactylation-directed strategies in clinical settings may significantly improve outcomes for patients with gynecological cancers. What are your thoughts on these innovative approaches? Do you think the potential benefits outweigh the risks? Share your perspective in the comments below.